Now, there are a number of factors that can alter enzymes, and I'd really emphasize this with you. This is very important. It has a lot of relevance to a range of things that we'll address over the coming weeks and into next term and next year as well. First of all, enzymes are proteins. We've got to remember that. And because they are proteins, this means that they can be denatured. By denatured, what we mean is that they can be broken down. So an enzyme, for example, might be amino acid joined to amino acid joined to amino acid, and so on. And there may be 20, 30, maybe even 100 of these amino acids all joined together to form the protein. If these get broken down into lots of individual amino acids, or perhaps even just simply smaller chains, that's what we mean by denaturing. And if that happens, the protein has obviously been changed, and therefore its action as an enzyme ceases. The types of things that can affect this, or the types of things that can cause enzymes to be altered in this way, include heat. So if you heat up an enzyme, it can cause it to denature and break down and therefore stop functioning. pH. This is very relevant in the digestive system because we have lots of enzymes acting in the digestive system, and the pH changes as you progress through the digestive system. Certain chemicals can cause enzymes to denature. Electricity and radiation are all things that can cause enzymes to be altered. Let's consider the effects of temperature and pH. Different enzymes have different optimal conditions. You'll find that some enzymes will operate in high temperatures, whereas others won't. Likewise, you'll find that some enzymes will work in a very acidic environment, whereas others prefer a basic environment. For example, digestive enzymes in the stomach. These ones are designed to operate optimally around a pH of 2. So the stomach is quite acidic. But when you get through to the small intestine, it becomes slightly basic. The enzymes that worked in the stomach will cease to work once they move through to the stomach, through to the small intestine. You need in the small intestine a different set of enzymes to digest the food. The same enzymes will not work because the pH has changed. In fact, the digestive system controls and manages what gets digested where by this fact. Here's a graph illustrating what I'm talking about. Here we have pepsin. Pepsin is one particular type of enzyme that functions in the stomach, and you'll notice here at a pH of 2, which is quite acidic, that its functioning is in fact at its peak, whereas you'll notice another enzyme, trypsin, here, its peak is at 8, a pH of 8. And neither of them actually overlap. So you don't get anywhere within the digestive system where both of them are actually working. Here's an example of temperature. Human enzymes function most effectively. Human enzymes function most effectively at body temperature. And of course body temperature is about 37 degrees. And as the temperature increases, the effectiveness of those enzymes increases up to that point. Beyond this, the effectiveness decreases. This is why fevers and high body temperatures are such a problem, because if your body temperature becomes too high, well then the activity of the enzymes of your metabolic processes is affected. Now on the other hand, some bacteria actually live in very hot thermal pools, and you can see here this is quite high, so we're talking probably about 75 degrees, and those enzymes can function quite happily in that environment. So therefore the organisms that contain those enzymes, bacteria, can live in those environments quite comfortably. Enzyme inhibition. Now You need enzymes to work in certain places at certain times, but you also need to stop them from working at certain times, and this is done through enzyme inhibition. Enzyme inhibition is performed by things called inhibitors, and what these inhibitors do is they essentially stop the enzyme from acting, and they do this in a range of ways. One of them is by changing the actual structure of the enzyme itself, changing the shape of the protein, and therefore changing the shape of the active site. They do this in a couple of ways. One is through what is called competitive inhibition, and the other is called non-competitive inhibition. And we need to understand that sometimes the effects of this are irreversible. So once you stop the enzyme from functioning, that's it, done. Let's consider competitive inhibition first of all. A competitive inhibitor is a chemical that has a very similar structure to the substrate. And because it has a very similar structure to the substrate, it is able to lock into the active site, and in doing so prevent the substrate from connecting to that active site. By doing so, it's effectively competing for the active site with the substrate. Okay, non-competitive inhibition. In this particular instance, the inhibitor does not have a structure like the substrate. And what it actually does is it connects itself to another part of the enzyme. And by connecting itself to another part of the enzyme, it distorts the shape of the enzyme, and in doing so, distorts the shape of the active site. It changes the active site. That means, therefore, that the substrate that would normally join with that enzyme no longer can. The active site shape has changed.